pathway for RNA Seq from single-cell or challenging samples. So today our speaker is Sarah Gonzalez, and she's a product manager at Takara Bio Europe and specialist in RNA Seq and NGS product line. So after obtaining a PhD from Lille University, Sarah joined the Pasteur Institute, where she studied degrading pathways of mRNA using next-gen sequencing and particularly RNA-seq. She joined Takara Bio Europe in 2014, where she is now a product manager and technical specialist for the next-generation sequencing product line. So with that, during this webinar, you will be muted. So if you have any question, you can ask by typing your question in the chat box at the lower part of the panel on your screen. So with that, I will leave the words to Sarah. So welcome, Sarah. Welcome to all, and to you, Sarah. Thank you.
Thank you, FX. So good morning, everybody. As FX said, today I will be talking to you about a smarter way for RNA-seq from single cell and other challenging samples. So I will start my presentation giving you an overview of our SMART technology, and then we'll show you how this technology has enabled to obtain a high-quality RNA-seq data from the most challenging samples. And I will be focusing in a single-cell mRNA-seq, in total RNA-seq from degraded and low-input RNA, and also in a small RNA-seq. At the end of the presentation, I will also explain a little how we have expanded the SMART technology also to DNA sequencing in another kind of particular challenging sample, which is chip DNA. So what is SMART? Because you will hear this along all the presentation. SMART stands for a switching mechanism at the 5' end of RNA template, and is based on two properties of MMLV, are reverse transcriptase, terminal transferase activity, and template switching activity. So how does it work, cDNA synthesis with SMART? First, we will start reverse transcription with a primer that can be oligo-GT primer or random primer, depending on the application, as we'll see later. And this primer will also carry this sequence in green that we call SMART sequence. So the reverse transcriptase, in this case we call it SMART scribe reverse transcriptase, will start the reverse transcription. And when it reaches the 5' end of the template RNA, it will add some non-template nucleotides. So this is the first activity, terminal transferase activity. And we have characterized this non-template nucleotides to design an oligo, shown here, called the smarter oligo, that will contain nucleotides that will anneal to this non-template stretch. And also it will carry another SMART sequence, also shown here in green. So what will happen next is that a smarter nucleotide will anneal here with the non-template nucleotides, and this will create an extended template to enable the RT to continue the replication. So this is the second activity, the template switching activity, because the RT will use now the smarter oligo as template instead of the RNA template. As a result, we will have a double-stranded cDNA after PCR, And for this PCR, we will use a primer that will bind the sequences that we have incorporated at both ends of the cDNA. So why using smarter technology for next generation sequencing? For the reasons that I showed, smarter cDNA synthesis provides exceptional sensitivity and also is very reproducible, as we will see later. So it allows cDNA synthesis from very limited amount of input. So it enables us to go to the single cell level. It maintains the original RNA length representation in the cDNA pool because this template switching activity will only occur when the reverse transcriptase reaches the 5' end of the transcript. Also, the method is very fast and streamlined, single tuber protocol, and this will greatly reduce the risk of RNA contamination or degradation. So for these reasons, smarter cDNA synthesis is currently the industry gold standard for ultra-low input and single-cell RNA-seq, and we will see how we have applied this technology to all these kind of challenging samples. But let's talk just a little before about transcriptome analysis with NGS, why people use this. As you probably know, RNA-seq will allow us to obtain the transcriptome of an organism, and this powerful approach has been used mostly for evaluating different gene expression between different conditions and cell types, but also to discover and annotate complete transcripts and to characterize alternative splicing isoforms, alternative polyadenylation sites, single nucleotide polymorphisms, etc. There's also a big interest in identifying non-coding RNAs that can be modulated, involved in the modulation of gene expression. So not only studying the messenger RNAs, but also these non-coding RNAs that can be divided in two categories, the long-coding RNAs and small RNAs. And finally, in these recent years, there's more and more interest in studying the transcriptome at the single cell level. And this is because it has been shown that the gene expression actually can vary very significantly between cells. And when we measure the expression of a population, we are obtaining an average signal that not necessarily will correlate with the gene expression at the level of each individual cell. And these cell-to-cell differences can be critical in physiological processes and also in disease. And if you are interested in reading more about single-cell RNA sequencing, there is this very nice recent review that you can consult. So talking about these differences, cell-to-cell difference important, I will just show here two examples of work done with single-cell transcriptomics. So in this paper, what authors found is that the splicing patterns can be very different between the cells. So if we see here the transcriptome profile of the bulk population, in this case 10,000 cells, for some specific genes, we'll see that there are two categories. There are the genes for which splicing pattern is very similar, and there are others that here, as you can see, have different patterns depending on if you are analyzing the different single cells. So if you only see the bulk, you will see a very constant pattern, but then in the single cell level there are differences. And also another study that investigated the single cell differences in a disease model is in this paper, in which glioblastoma tumors were dissected and dissociated, and then single cell sequencing was performed on the different cells, and actually the authors found that cells have different transcriptome profiles depending on the region of the tumor that they are located, and this has implications also in the phenotype of the tumor cells. So doing transcriptome analysis from single cell is very challenging because of the size of the input material, the fact that it's not really possible to extract the RNA at the level of a single cell. So what we have developed with our smarter ultra-low technology and with the current SmartSeq V4 technology is a solution to enable directly the synthesis of cDNA from the cells without doing RNA extraction. So this kit allows to work directly from cells, as I said, from 1 to 1000 cells. Of course, if you want to purify RNA and start from purified total RNA, it's possible. So it has a sensitivity range of from only 10 picogram and up to 10 nanogram. And this is because we estimate that the RNA content in one cell is roughly 10 picogram. So the smart ultra-low technology uses oligo-DT priming to specifically amplify messenger RNA, and also this allows us to avoid the ribosomal RNA removal or DNase treatment. So we can see here the protocol for smart ultra-low synthesis, which is very similar to what I explained before for the smart reaction. And so it's a single tube protocol with minimal handling, and at the end we will obtain a double-stranded cDNA and full-length cDNA, as we can see here from this bioanalyzer profile showing the different profiles from different input amount. So from 10 picogram to 1 nanogram, in all cases we will obtain full-length cDNA. So it's important to note that a SmartSeq V4 allows to do the cDNA synthesis, as I said, but after this cDNA needs to be processed for adding the corresponding adapters for sequencing, and also, as is again full length, it needs to be cut, because you know that sequencers require smaller fragments to for cluster generation. So the cDNA generated is compatible with both ion torrent or Illumina preparation. If you are using ion torrent, you can follow a protocol that we provide in the user manual for doing enzymatic sharing, and using also the Thermo Fisher Ion Express library prep products. If you are using Xterra, you have two options. One is using Xterra XT, that maybe you are familiarized with. So here we say it's a modified protocol because only because you need to use actually less input than Xterra XT recommends, which is they recommend 1 nanogram at least to start. If you are using the SmartSeq V4 cDNA, we recommend to start with only 100 picogram for better results. But the rest of the protocol is exactly the same. Or you can also do physical sharing of the full-length cDNA using Covaris, and then use our low input library prep kit to add the Illumina adapters and ultimately proceed to sequencing. So you have noticed that the name of the kit is SmartSeq v4, which implies that we had previously other versions. And actually, since the first version of the Smart Ultralow technology, we have constantly added modifications and improvements to get the best results. So as compared to the previous version, the Ultralow v3, we have made improvements to the template switching oligo, and namely we have licensed this incorporation of LNA, which stands for log nucleic acid, from the SmartSeq 2 protocol, which is a homebrew method developed by the Karolinska Institute. And this LNA has as feature to provide more sensitivity. So we have incorporated this LNA and also build upon our long experience with modifications to the template switch oligo to reduce the background and significantly improve sensitivity and reproducibility in the current version. So how we can see that? We can see that here in this comparison, in which we are showing results for two different replicates, technical replicates, all done with 10 picogram of mouse brain RNA. So we use exactly the same input just to measure the technical variability between the different libraries. And what we can see here first is that SmartSeq v4 is the sensitivity, as it has been increased. It allows us to use less PCR cycles, and yet we can obtain the highest number of transcripts as compared to our previous v3 version or the SmartSeq 2 protocol. Also, something that can happen occasionally with the SmartSeq 2 protocol is that the map to the genome could be less important, and this is because there is sometimes a phenomenon of concatenars that are observed, and it reduces the amount of reads that map to the genome. With the SmartSeq v4, as with the previous version, this the mapping to the genome is very high and very reproducible, and the modifications that we did to the template switching oligo also allows to not having this problem of concatenarization. Another way to look to the increased reproducibility and sensitivity of the SmartSeq v4 is these scatter plots, in which we are comparing two technical replicates again, and we are representing the expression of each transcript in each replicate as a dot. So the more the line looks diagonal, the more the two replicates are similar. And also this can be measured for these coefficients of correlation that should be ideally close to 1. What we can see here with the SmartSeq v4 and also with the other technologies is in the, if we analyze the higher expressed genes, the reproducibility is quite good. But if we focus in this lower part, which corresponds to the less or the lower expressed genes, we can see that there is it looks more scattered, so it's less reproducible. And also, if we see the coefficient of correlation, we can see that only with SmartSeq v4 we obtain values that are higher than 0.5. So this shows that reproducibility and sensitivity is higher with SmartSeq V4. We have also done studies using HeLa cells, obtaining the profile of a single cell. As you can see here, these are the bioanalyzer profiles. We obtain full-length cDNA, and this is for 6 different single cells, and this is for bulk population. The quantities are not the same, they vary in between cells, but the profile is very similar. Also, when I said before that the smart technology allows to maintain the full-length body coverage, we can see here this. We have compared the body coverage across different input amounts, and we can see that it is very uniform and well distributed from 5' to 3'. Of course, I show you here data with HeLa cells, but it's more interesting to know what this has been done like with more interesting samples for researchers. So here we have put just some of the data and the single cell studies that have been done using smart technology. So you can see that it has been applied to many different cell types and for many different applications, and we have many more references available. For this work, people use different methods to get the actual single cell. You can just do micromanipulation, do it the manual way by serial dilution. You can also use FACS to sort the cells, or laser capture microdissection, and also some other have used immunocapture techniques using beads coated with antibodies targeting specific cell surface marker. And there is also some automated instruments like the Fluidigm C1. So this instrument, which is developed for by another company, Fluidigm, allows to automatically capture the cells from a suspension and distribute it in different wells by using the microfluidic technology. And it will also allow to inject the reagents necessary for lyse, reverse transcription, and PCR. And then after that you will do your library preparation using Xterra as per Fluidigm recommendations. But what's interesting here is that the reagents that are used for this single cell cDNA amplification are actually smart reagents. And for a long time we have the very first version of the kit for the Fluidigm machine, and very recently we have launched the newest version which incorporates the V4 chemistry for this machine. So this is the SmartSeq V4 kit for the Fluidigm C1 system. So now moving from single cells to other kind of challenging samples, and I'm thinking about degraded low input RNA, so kind of RNA that you can get from FFP samples, for example, that can be, because as I said previously, the SmartSeq V4 uses oligo-DT priming. It means that you need to have a good quality RNA to ensure that you have enough polyadenylated material to prime transcription. But if you don't have this because your RNA is degraded, or also because maybe your organism of interest is not polyadenylated, you need to use random priming. So we have developed a method, technology called Smart Stranded RNA Seq Kit that will use random priming but based on the same smart technology. And this allows us to use the technology with RNA samples of any quality. We have measured that it can be used from samples with the ring 3 to 10. It can be used with a very low input amount, so from only 100 picograms to 100 nanogram of ribosomal RNA depleted RNA, and for this we have also a solution, as I'll say later. And an important feature of this kit is that we have adapted the smart technology to incorporate in the same time during RT and PCR the Illumina indexes and adapters required for sequencing. So the advantage of this is here you will get directly a library ready to sequence. You can go from your RNA to a library in only 4 hours. This is very fast. But stranded RNA Seq kit is not only used when you have degraded RNA, you can also use with good quality RNA because, as I said, it's compatible. And you may also be interested to use it if you are studying both coding and encoding RNA. If you are working with non-polyadenylated species, as prokaryotic, and also if you're interested in studying antisense RNA, because the kit provides inherently strand information. As you can see here, you will be able to identify the reads from each strand. So this is also an advantage of this technology. As I said, one of the limitations that we can have with total RNA-seq is that most of the RNA in a cell is total RNA, ribosomal RNA, and this needs to be depleted to not to waste sequencing in this ribosomal RNA. We have developed a solution compatible with the low input range of our total RNA-seq kit. This is Ribogon mammalian, which is compatible with only 10 nanogram of RNA, and also can use RNA of any quality from ring to 10. This is mammalian specific kit. Here we can see deficient RNA, ribosomal RNA depletion using Ribogon, and then preparing the library with the smarter stranded RNA-seq kit. You will see that the percentage of reads that align to ribosomal RNA is very low, and we have very good mapping to the genome and exonic and intronic regions. Again, we have measured the reproducibility of the kit and comparing here across the lowest input amount along with the kit and the highest input amount, and you can see that the correlation is very high. So this is useful if you are analyzing samples that you have different input amounts. You can be confident that you are detecting and that you can compare your different input amount samples. Here, I want to just briefly mention that if you have a normal amount of RNA, but if you like the rapidity and the simplicity of this workflow, we have developed a solution which is compatible with high input. What we call high input is 100 nanogram to 1 microgram of total mammalian RNA, and also compatible with RNA of any quality, and you will have this advantage of having a very fast method, because you will be able, to go from your total RNA sample, and the built-in Ribogon technology for RNA removal, ribosomal RNA removal is included, and you will obtain your library ready to sequence in 5 hours. So getting back to challenging samples, what happens if you have pico amounts of RNA and is so small that you cannot use ribogon or other ribosomal RNA depletion method, because remember, with the ribogon you need at least 10 nanogram, and most of other kits available out there require at least 100 nanogram. So we have developed a method very innovative that allows us to deal with these samples. So with the smarter stranded total RNA Seq kit pico input mammalian, you will be able to start from only 250 picogram of total RNA and up to 10 nanogram. And still you will be able to deplete your ribosomal RNA sequences. So how is this possible? This method is different from what we have been used so far, because we will do the cDNA synthesis from all the material, the input material, including the ribosomal RNA shown here in purple. And the protocol and smart reaction is very similar. But then we will do the depletion of the ribosomal RNA sequences at the cDNA level using a proprietary technology that we call as ZAP-R and R-Pros that will target specifically the cDNA derived from ribosomal RNA shown here in purple. As a result, and after a step of enrichment by PCR, we will obtain only the sequences of interest. So this method allows, again, to obtain a ready-to-sequence Illumina library, which provides the strand information and is very fast, again, 5 hours. And most importantly, for very small amounts of total RNA. So everything is included here. Here I just want to show you some comparison data using the different amounts of RNA. So I say that the range of the kit is 250 picograms, but here we show that it's possible to go a little below with 100 picogram. Of course, it will depend on your sample. It may be not possible for all samples. You could also compromise some sequencing quality data if you go too low. But what we can see here is, regardless of the input amount used, the number of transcripts identified is very similar. The strandness is very good. And you will also note that ribosomal RNA left is higher as compared that I showed before with ribogon. Here we are around 10%, with ribogon is more 1%. But still, this is very good, and as compared to the 90% that you will probably obtain if you didn't do the ribosomal depletion. We have also measured the reproducibility across the recommended input range, and this with two different kind of samples. So here with a nice normal sample, mouse brain total RNA of high quality, but also with degraded sample. This is a FFP sample from a human liver total RNA. And you can see that in both cases, the reproducibility between the lowest input amount and the highest input amount is very good. Then we also wanted to show here that the kit efficiently allows to capture RNAs of small size, so at least 100 nucleotides that we show here. Again, comparing the two samples, the sample here in purple is a RIN sample with a FFP sample with a RIN of 2.5. And in blue, you can see a sample of good quality with a RIN more than 8. So the protocol, it includes actually a sharing step that needs to be used or not depending on the quality. So for high quality, we did the sharing step. For RNA, which is already bad quality and fragmented, there's no need. But in both cases, RNAs are efficiently incorporated. But what happens if you are interested in studying really small RNAs? So your RNAs are less than 100 nucleotides because you are studying microRNAs, PUE-RNAs, and so on. So this is another challenging sample because typically it's difficult to capture these very small RNAs in GS protocol. Another challenge in small RNA sequencing is that most methods are based in ligation, and as you may know, ligation is subject to bias because not all sequences get ligated with the same frequency. And another limitation of a small RNA seq is that most of protocols use a gel-based method to enrich or to separate the small RNA fraction. So with this new method that we propose here for small RNA sequencing called Smart Small RNA Seq for Illumina, we wanted to provide a ligation-free Illumina library method. So again, based on our SMART technology. As you can see here, what we will do is just polydenylate the small RNA, and then using our oligo-DT priming, we will proceed to the SMART reaction, and we will be able to add the known sequences at each end of a small RNA. So this is usually done with ligation, put the adapters in each end. We do it with our SMART reaction. The advantage of this also is that we can go a very low input amount, so we can start from only 1 nanogram to 2 microgram of input RNA. So this is the lowest input amount currently possible in the market. And the input is very versatile because you can start from total RNA or also from enriched fraction, because some protocols require you to start only with the purified fraction. You can use it to create libraries from small RNAs from 15 to 150 nucleotides, so including microRNAs, siRNAs, pRNAs, etc. But also you can use it for other applications that may be generating RNAs that are very fragmented and lower than 100 nucleotides that couldn't be captured with the PICO kit, for example. And this we can think to in applications like RIP-seq, CLIP-seq, or ribosomal profiling. We, as in all our kits, we ensure that reproducibility is good, first comparing here two technical replicates, and also across the small input amount and the biggest input amount. In both cases, correlation is very good. And here we wanted to show a comparison with the ligation method. So for this, we use a synthetic library which contains more than 900 microRNAs, which are exactly at the same equimolar concentration. And it means that if given that they are in the same quantities, each should be represented equally in the sequence output ideally. Of course, what we see after that is that adapter ligation showed a bias because only 22% of the mRNAs were found in the expected cutoff level. So if all had the same expression, we will expect them to be around a cutoff of 1, but with the adapter ligation method, only 22% fall in this range. With smarter technology, we have significantly improved this. We found that more than the half are in the expected cutoff range. So this shows that we, with the smarter approach, are more accurately portraying the expression of the mRNA species as compared to ligation. Okay, so we are into the end of the presentation. I want to finish just saying briefly how we have expanding this smart technology, which is very versatile, to other challenging samples. So I will just briefly talk about this application for chip seq samples. And just for you to know, that we have also applied smart technology to high throughput profiling of the T-cell receptor repertoire. This is a very specific kit, the Smart Human TCR profiling kit. And I won't talk about this one today because we are having a webinar on 9th June, so I invite you to register if you're interested in knowing more about this. So talking about chip is also a challenging sample, actually, chip DNA, because usually what you obtain from chip is very low amounts of DNA, and also it can be single-stranded or double-stranded. This can be an issue. Some protocols absolutely require double-stranded DNA. So we have discovered that actually the reverse transcriptase that we use in smarter protocols can also use a single-stranded DNA as template. So we have adapted the technology to be able to produce ligation-free Illumina libraries from the input single-stranded or double-stranded DNA obtained with chip. So this is very fast protocol, only 4 hours to get your library. And to make things faster, we have also a chip elute reagent that will allow you to reverse, cross-link, and purify the immunoprecipitated DNA in only 1 hour. This is significantly faster than other protocols that can take up to overnight. Here, just to show you some data that we have obtained, you can go with as low as 10,000 cells or more if you want, but in any case, the results are very reproducible, and the overlap between very small input amount or high input amount is very good. Of course, this will also highly depend on your antibody. Okay, so I hope that this presentation could show you how smart cDNA synthesis can be used for all these challenging samples that we have discussed, thanks to the exceptional sensitivity and reproducibility. Also, I just want to show this RNA selection guide, which summarizes all the technologies that I have presented here. This is available on our website and will allow you to choose the appropriate technology depending on your input amount, the kind of sample, the type of RNA that you are wanted to do. And for each of our products, we have dedicated webinars and tech notes, and you can go and see this in the NGS resource portal. And of course, if you have questions, do not hesitate to send it to our tech email address. And that's good science. Thank you for your attention. And now, if you have questions, we'll be happy to take them.
Thank you very much, Sarah, for this nice presentation about the challenging samples that can be addressed with the smarter technology. And there are a few questions actually, and I will take them through the chronologic so as they arrived. So the first question was, does the smarter stranded RNA Seq kit also work for cell-free plasma, small RNA, microRNA, and PW RNA?
Yes, so I guess that the question came before I talk about the small RNA-seq kit. Actually, the standard RNA-seq kit is recommended only for RNAs that are more than 150 nucleotides. The Pico kit can capture more than 100 nucleotides, and for everything that is below, so small RNA-seq kits including microRNA and PW RNA, we recommend the small RNA-seq kit.
Okay, thank you. And the next question was, does the smarter stranded RNA-seq kit provide uniform gene coverage?
Yes, it does. I didn't show the data here, but if you go to our tech notes that are in the website, we have also gene body coverage, similar results that what we observed for what I showed for the SmartSeq V4 mRNA-seq kit.
Okay, so no bias 3' or 5'.
No, even body gene coverage.
Okay. Does the technology, so I guess it's generally for the smarter technology, does this technology have been applied to a single cell from plants?
So for what can be difficult with plants is that they have the cell wall and might be difficult to lyse with because actually in the SmartSeq V4 we include a lysis reagent buffer that will allow you to get access to the cell material. And we have not tested that in-house with plant RNA. So depending on, with plant cells, depending on your cell type, it could work, but we have not tested it. We think that if the cell wall is too important, it may be not strong enough, the lysis buffer, to lyse plant cells.
Okay, thank you.
But
So
sorry, I just
work.
want to add that it can be used for plant material. If you isolate RNA, it can of course be used.
Okay, very good. So now a question about the small RNA seq kits. Since the polyadenylation is not sequence specific, will this method also generate other non-specific small RNA fragments like microRNA or pRNA, so which may make things difficult to find the proper small RNA or the small RNA of interest?
So indeed, the polyadenylation step will polyadenylate everything that is there, and also the oligo-DT primate can prime some other like messengers RNA that are in the sample. That's true, but the other steps of the protocol are optimized to only favorize the amplification of the small fraction. And the PCR step is very fast, it's 5 to 10 seconds of elongation. So you are really not favorizing the amplification of smaller fragments. And then after that, you get this cDNA, there's a step for size selection. You need to size select for the small fraction to enrich it. This can be done using blue pipping or pure base size selection. But no, as I say, what is nice in this protocol also is that the two methods are not based on just going and cutting your gel, which can be very tricky and is normally what is used.
Mm-hmm. Okay, thank you. Then there's a question about if we have a kit for RNA-seq on blood samples.
At the moment, no. I guess that you are. I mean, you can use this with, for example, the stranded RNA seq with blood sample. The issue with blood sample is they usually need to use globin mRNA depletion because it's a very abundant transcript. So if the question is that we have something specific to deplete globin mRNA, no, we don't. But there are other commercial solutions to doing that, and after that you can totally use the depleted material with any of our products.
Okay. And do you know if it's in development or something?
We hope to have something, but for the moment, I will not be able to say when.
Okay, thank you. Then there was a question about the ribogon. So, do you develop kits to remove specific ribosomal RNA, not only mammalian? So.
We are working actively on that, especially for getting a ribogon that will be able to work with prokaryotic samples. So hopefully we'll have it in the near future. At the moment, yes, it's only mammalian.
Okay, thank you. And then there was a question about long non-coding RNA. So how would you obtain how would you obtain the sequence of long non-coding RNA that do not have a poly-A tail?
You can use smarter stranded kit, definitely, because it's random priming, so no need to having a poly tail. And also, as many long non-coding RNAs are expressed in antisense orientation from coding transcripts, it will also be very useful, the smarter stranded kit, because you will be able to differentiate the expression from both the sense and the antisense strand.
Mm-hmm. Okay, thank you. And then a question about the workflow of the small RNA seq kit. So how long does it take to perform this workflow?
Yes. Also, another thing that another challenge with current small RNA seq techniques is that these are very long and multi-step protocols. With our protocols, we want also to make things easier for customers, and actually the first steps, so the actual cDNA synthesis from small RNA is very fast. It only takes 3 hours, and then you will have the other size selection steps that can take a little longer, but you can complete the protocol in 1 working day, the whole protocol.
Okay. So there are a lot of questions, Sarah. So the next, I lost track. Because they are all arriving in batch. Thank you. So do you have any plans to develop a smarter stranded RNA seq kit for plant samples?
So for plant samples, you can use a smarter stranded RNA seq kit. The difficulty again is the ribosomal RNA depletion method. Ribogon for plant for the moment is not something that we have, but you can use, there are other ribosomal depletion methods, commercial methods, to deplete RNA from plant. You can use this to deplete your plant material, and then use the depleted material as input for the smarter stranded RNA seq kit. And as long as you have 100 picogram of depleted material, it can work perfectly.
Okay, so basically, yeah, the problem here is still the depletion of
yes.
ribosomal RNA.
Yes, the problem is the technology for depletion, that we don't have it for plants, but then all the library preparation is definitely compatible with the smarter stranded.
Okay, very good. So maybe the last question, because it's already 11:30. So in a small RNA-seq kit, if the non-specific polyadenylated fragments are very small, 25 to 30 nucleotides, there will be no way to separate these fragments from microRNA and piRNA, correct? So this could be problematic in samples such as plasma, where you have highly fragmented RNA. So what would you recommend here, or what would you comment on that?
So I would say that if you have fragmented RNA that corresponds to the size close to your microRNA and piRNA, indeed you will not be able to separate this from the library, but I think that it will be a limitation with any preparation method, because everything is based on size. So if you are using, even if you use a protocol to specifically enrich a small fraction, everything that it will be in that size will be enriched. So I think that then is more than the analysis that you can do with the data. When you will just compare your data with the appropriate database, you will be able to see which is the fraction of your reads that will align with the microRNA databases.
Mm-hmm. Okay, so very good. Thank you again very much for this very nice presentation, and thank you all for your very good questions. I hope we gave you replies, and again, if you need to contact us, tech@takara-clonetech.eu, and we will reply to any further questions. So thank you very much, and see you soon.
Thank you very much for everybody for coming. Bye.